Welcome to my primer video in epigenetics. My name is Efi Kostarelli and I'm a lecturer and research group leader in epigenetics at Queen's University Belfast. I hope that this video will help you to prepare for the specialized lecture that will follow. By the end of this video, you will be able to provide the definition of epigenetics, describe the structure and function of nucleosome, compare and contrast heterochromatin and euchromatin, and name the mechanisms of the epigenetic machinery. Epigenetics is the study of heritable phenotype changes that are not attributed to alterations of the DNA sequence. The Greek prefix epi means above, on the top of, and implies features that are in addition to the traditional genetic basis for inheritance. Epigenetic mechanisms regulate gene function and define which genes will be activated and which silenced. This regulation is mainly achieved by chemical modifications to DNA or to histones. But how DNA and histones are structurally organized in the cell nucleus? DNA is compacted into chromatin together with histones, and the basic unit of chromatin is the nucleosome. Nucleosomes are composed of 146 BP of DNA coiled roughly twice around a histone octamer. The octamer consists of two copies of each of the four core histone proteins, H2, 2B, 3 and 4. Adjacent nucleosomes are joined by a stretch of free DNA termed linker DNA, which is associated with histone H1. The repeating structure of nucleosome is what results in the visualization of DNA as bits on a string under the microscope. But what is the function of nucleosome? Actually, it represents the first order of DNA packing in the nucleus and determines DNA accessibility. Based on DNA accessibility and level of compaction, we traditionally classify chromatin into two forms, euchromatin and heterochromatin. The major difference between the two forms of chromatin are the following. Heterochromatin is tightly packed, dark stained, and characterized by high DNA density as opposed to euchromatin. It is found only in eukaryotes, it is late replicative, genetically inactive, and exhibits little or no transcriptional activity. Heterochromatin has important functions such as gene silencing and structural integrity of the genome. Importantly, heterochromatin and euchromatin are marked by different combinations of epigenetic marks. The four mechanisms that are mediating DNA accessibility and gene expression are the methylation of DNA, the histone post-translational modifications, the ATP-dependent remodeling of chromatin, and the production of non-coding RNAs. Here we will discuss shortly the first two. DNA methylation usually occurs at the fifth position of DNA cytosine in the CpG islands located within the promoter regions of specific genes. Such a modification inhibits both the binding of transcription factors to DNA and affects the recruitment of proteins involved in chromatin remodeling. Thus, DNA methylation can cause gene silencing. Histone post-translational modifications are covalent modification of histones by phosphorylation on serine or threonine residues, methylation on lysine or arginine, acetylation of lysines, ubiquitinylation and sumylation of lysines. Histone modifications affect chromosome structure and function, for instance during transcription and chromatin remodeling processes. So I hope that you are now able to provide the definition of epigenetics, to describe the structure and function of nucleosome, to compare and contrast heterochromatin and euchromatin, and to name the mechanisms of the epigenetic machinery. Thank you very much for your attention.